Good afternoon, and welcome to the Brain and Behavior Research Foundation's Meet the Scientist monthly webinar series. I'm Dr. Jeff Borenstein, BBRF's President and CEO. Today, Dr. Bridget Callaghan will present Intergenerational Effects of Adversity on Mind-Body Health, Pathways Through the Gut-Brain Axis. BBRF is the world's largest private funder of mental health research grants, supporting transformative discoveries in order to develop improved treatments, cures, and methods of prevention. The high quality of the research we fund is made possible by the BBRF Scientific Council. This group of 195 prominent mental health researchers reviews each grant application and selects the most promising ideas with the greatest potential to lead to breakthroughs. The Scientific Council guides the Foundation to fund creative and impactful basic, translational, and clinical research relevant to the whole spectrum of mental health. One reason that research funded by BBRF has such a great impact is because we do not limit ourselves to one illness or condition. Since 1987, the Foundation has awarded $461 million to fund more than 6,500 research grants around the world and across a broad spectrum of brain illnesses, including addiction, ADHD, anxiety, autism, bipolar disorder, borderline personality disorder, depression, eating disorders, OCD, post-traumatic stress, schizophrenia, as well as suicide prevention. Today, I'm delighted to introduce Dr. Bridget Callaghan. Dr. Callaghan is an assistant professor in the Department of Psychology at the University of California, Los Angeles. At UCLA, Dr. Callaghan directs the Brain and Body Lab in the Department of Psychology, and she also heads the Mental Disorders and Pain Research theme at the Goldman Luskin Microbiome Center. She was a 2016 Young Investigator. Our webinar will begin with Dr. Callaghan's presentation, which will then be followed by Q&A. To submit your questions, please use the Questions tab on the control panel on your screen. Please feel free to submit your questions at any time. Following the presentation, I'll ask as many questions as possible in the time allotted. And now I'm pleased to introduce Dr. Callaghan. Bridget, the floor is yours.
Thank you so much, Dr. Borenstein. That's such a nice introduction. I'm really happy to be here today. I want to make sure that you can see the right screen.
Yes, indeed.
Perfect. Okay, great. So, oops, what's happened? Okay, perfect. So, yeah, I'm very happy to be here today to present my research on the intergenerational impacts of adversity on mind-body health. So looking at pathways that intersect with the brain-gut axis. I do want to mention at the start that, as Dr. Borenstein said, I was a recipient of the Young Investigator grant in 2016. And that grant was really fundamental in helping me set up a lot of the research that I'm going to present to you today. So my research program is really centrally focused on this relationship here, the attachment relationship between a parent and a child. A relationship that, when it is secure and strong, can really help children deal with all manner of challenges in this world. And when it is disrupted or ruptured, can predispose individuals to exhibit challenges basically in all different areas of their life. So we call adversities that happen within this relationship caregiving related adversities. And these include things such as parental abuse, neglect, extended separation from a caregiver. For instance, we know that disruptions to this caregiving relationship, so caregiving related adversities, are really heavily associated with mental illnesses. So such adversities account for up to 30% of adult mental illnesses, making them one of the most reliable risk factors for mental illnesses that we're aware of. And not only do they increase the incidence of mental illnesses, but we also know that they increase the incidence of nearly all examined physical health problems, including things like heart disease, respiratory disease, and gastrointestinal conditions as well, like irritable bowel syndrome. Now, critically, these risks for worsened physical and mental illness following caregiving-related adversities really don't stop with the individuals who were directly exposed, but they can also be perpetuated across generations. And in fact, there's now a lot of evidence for the perpetuation of early adversities across generations. Now, this has been seen most famously at the population level, where children of individuals who experience things like starvation or war or genocide themselves go on to experience or at risk of experiencing worsened physical and mental health, despite having not been exposed directly to those events themselves. But we also see this perpetuation playing out in really smaller studies after caregiving-related adversities. So given this information, it really highlights for me that there are two major priorities that we should have right now in our understanding of well-being. And the first is that we really need to be thinking transdiagnostically about mechanisms that can explain the diversity of symptom profiles that are observed after adversity exposure. And that includes within mental health, so the coincidence of anxiety and depression, for example, they're very highly comorbid, as well as between mental and physical health. So for instance, we know that irritable bowel syndrome and anxiety disorders tend to go hand in hand. The second thing that we really need to understand is that we need to really appreciate the importance of these generational experiences and mechanisms that could potentially account for the transmission of risk across generations after adversity exposure. And so having both of these foci in place will hopefully get us to an area or a space where we can better address the multitude of adverse outcomes which are attributable to adversity exposure. Fabulous. All right, let's zoom through and get to where we were at. Okay. So sorry about that, everyone. So in terms of really focusing on those transdiagnostic mechanisms, because remember I was talking about these two research priorities we should have, one being these transdiagnostic mechanisms and the other being intergenerational patterns that could explain intergenerational transmission of risk. In terms of the transdiagnostic mechanisms, I have a particularly strong interest in the brain-gut microbiome axis because of its known associations with mental and physical health across really a range of diagnoses and domains. Now, this axis is a communication highway that's actually bidirectional. So there's communication from the brain to the gut and the gut to the brain. And this communication occurs through multiple different pathways, including through the vagus nerve, as well as the spinal cord. And it also involves a very heavy role for chemical signaling. And one of the reasons that I'm so interested in this axis is because it is transdiagnostically implicated in a range of different disorders, including things like depression, anxiety, cardiovascular disease, irritable bowel syndrome. Really, you name it, there is a role for the gut microbiome in it. So it's very important from that diagnostic perspective. And then finally, for the purposes of the talk today, the brain-gut microbiome axis does seem to be affected by early life caregiving adversity, and there are a range of different studies highlighting that to be true, coming out now. So what I'm going to do today in this talk is to do it in 4 parts. So in the first part of the talk, I want to show you data that spans just one generation. And I want to ask the question, how does early caregiving adversity affect brain development, and how is that associated with youth's mental health problems? Sticking within that first generation, I then want to move into part 2 of the talk, where I'm going to ask how early caregiving adversity affects the microbiome and brain-gut axis in ways that are important for mental health in youth. In part 3, we're going to begin to take that more transgenerational focus. So asking how early caregiving adversity in generation 1 affects brain behavior and the brain-gut microbiome axis in the next generation, generation 2. And then finally, in the 4th part of the talk, I want to ask how early caregiving adversity affects the way that one transitions into motherhood and the risk for perinatal depression. And the reason for that is that perinatal depression, we know, is really one major factor involved in intergenerational cycles of adversity. So thinking about how these early life experiences that a mother has influences peripartum depression would be really important for that generational piece. All right, so let's dive into the first part of the talk today, looking at the impacts of caregiving adversity on brain function in youth. So what I'm going to focus on here is the impacts of early life adversity on one brain structure, its functionality, called the amygdala. Now, the amygdala is this small almond-shaped region right here. And the reason that I'm particularly interested in the amygdala in my own research is that numerous studies have shown that caregiving adversity has an outsized impact on limbic system development, including on the amygdala. And this is likely because these regions undergo rapid structural growth during the early life period when children are most dependent on their parents for affect regulation, their emotional regulation, and their emotional life in general. So if these brain regions are going to be responsive to any environmental cues to guide their development, then caregiving adversity is very likely to be one such cue of importance for the development of these brain regions. Now, indeed, numerous studies have shown that the amygdala is particularly responsive to caregiving cues in the environment. So this effect was actually shown in a study by Dylan G back in 2014. And what she did was to have children lie in a scanner and look at pictures of their parents' face or a stranger's face. And the strangers were age and sex and race matched to the parent. And then what she did was to simply contrast amygdala activity. So how active is the amygdala when looking at pictures of the parent's face versus the stranger's face? And then she contrasted that activity. And what she showed was that specifically during childhood, so in the gray bars here, there was lower amygdala reactivity in children to pictures of the parent's face relative to the stranger's face. And this effect was absent in the adolescent period of life. And so they really interpreted this finding as the amygdala buffering or decreasing its activity to parent cues in childhood. But then as the child turns into an adolescent, as they become increasingly independent from their parent or caregiver, then the amygdala becomes less responsive to those parent cues. Now, if that's true, then we hypothesize that a lack of caregiving cues in the environment from early in life may begin to affect what the amygdala reacts to and may decrease the amygdala's reactivity to parent cues even earlier in development. So I actually did my training in this same lab, so in the lab of Dr. Nim Tottenham, who's at Columbia University. And the first thing I wanted to do was to ask how exposure to caregiving related adversities affected amygdala's responsivity to parent cues in childhood. So I did this exact same task, having children lay in the scanner and look at pictures of their caregiver or a stranger in a group of previously institutionalized youth. So these youth are raised in institutions or orphanages overseas before them being adopted into American families. And this is an extreme type of caregiver deprivation early in life, which has been shown to increase the risk for internalizing symptoms, so anxiety and depression. Now, as you can see, these previously institutionalized youth, the children, did not show that amygdala buffering effect. In fact, their amygdala profile actually much more closely paralleled the adolescents of the comparison group. And this was not associated with age of adoption or the amount of time that they had spent with that adoptive caregiver. So this suggests that caregiving adversity disrupts the responsiveness of the amygdala to parent cues in childhood. Moreover, individual differences in this effect, and you can see through the size of these error bars, this illustrates the variability in the sample. This is quite a large error bar, so there was a lot of variability in responses, and this variability was clinically significant or clinically relevant. So what do I mean by that? So here we've got, I'm plotting anxiety on the y-axis, and we've got 2 years of the study. So year 1 is at the same time as the MRI scan, and year 3 was 2 years later. And I've got the comparison group on the left, the adversity group on the right. And I'm splitting the data into individuals who showed that amygdala buffering effect, so lower amygdala reactivity to pictures of the parent than the stranger, and individuals who did not show that buffering effect. So more of the adolescent profile and more of the child profile within the comparison group. What we see is within the comparison group, the buffering doesn't make too much of an impact on internalizing symptoms. Internalizing symptoms tend to be rather low in everyone. But within the early life adversity exposed group, so previously institutionalized youth, those individuals who exhibited parental buffering of the amygdala showed a sharper decline in anxiety from year 1 into year 3 of the study. Moreover, it was shown, or we showed, that attachment security was associated with this effect. So these are previously institutionalized youth who showed parental buffering of the amygdala at time 1, and they self-reported feeling more secure in their attachment relationship with their adoptive caregivers than individuals who did not show that amygdala buffering effect at time 1. So this suggests to us that individual differences in amygdala buffering to parent cues are relevant for affective functioning across development, and particularly for anxiety. And that's important because this could potentially be targeted through parenting interventions. We have ways to increase children's felt security in attachment relationships with caregivers, biological or adoptive. Now, in another study, we've shown that while parent cues no longer buffer or decrease the reactivity of the amygdala in adolescence, parent-adolescent interactions still do shape amygdala connectivity patterns in ways that are associated with adolescents' mental health, even years on down the line. So this is a 7-year longitudinal study that I did in collaboration with Dr. Sarah Whittle's group at the University of Melbourne in Australia. And what we did in this study was look at how aggressive parent-adolescent interactions when youth were 12 years of age were associated with their brain connectivity, so specifically the amygdala, how it connects with other brain regions when those youth were 16 years of age. And then finally, whether that mediated the association or helped to explain the reason for the association between these aggressive parent-adolescent interactions early in adolescence and then late adolescence, first onset diagnoses of major depressive disorder at 19 years of age. What we found in this study was that maternal aggression at 12 years of age was related to altered resting state. So at rest, kids are laying in the scanner, their functional connectivity between the amygdala and a range of different brain regions, most notably the temporal cortex and some of the insular cortex here circled in red. And it was amygdala connectivity with this region here circled in red that connected maternal aggression towards the adolescent at 12 years of age, and then adolescent first onset depression diagnoses at 19 years of age. So again, parent-child interactions from early in life all the way through adolescence seem to shape amygdala circuits in ways that affect later emotion regulation and mental health. And there are clear ideas here for intervention. So more positive parent-child relationships may have a real impact on children's health, and we think that this may operate in part through the effects of that relationship on children's amygdala development. All right, so far in part 1 of the talk, we've looked at the type of early life experiences, so caregiving-related adversity, and its impacts on the brain. But one underexplored possibility is that caregiving-related early adversities may increase youth risk for psychopathology through a more widespread impact on both central, so that is the brain, as well as peripheral systems, including the gut microbiome and the wider brain-gut microbiome axis. So in part 2 of the talk, I'm going to focus on the impacts of caregiving adversity on that wider brain-gut axis. So as I mentioned at the start of this talk, several studies have already shown that early life adversity is associated with changes to the microbiome. However, most of those studies, including many of my own, have been kind of early in the piece. So they've used small sample sizes, or they've had adults who've retrospectively reported on their experience with early life adversity. Moreover, all of the literature focusing on the impacts of adversity on the microbiome have focused on one microbial community, and there are many in the body. So they're focused on the gut, and that's to the neglect of other microbial communities, for example, the oral microbiome in our mouth, that may play and seem to play an important role in our mental health. So in this next section of the talk, I want to show you what we're discovering about how adversity at different stages of development affects the microbiome and how that then links to youth's mental health outcomes. So firstly, I want to familiarize you with some metrics of the microbiota. So I'm going to focus on 2 primary metrics today. The first is alpha diversity. This is a within individual metric where we take the bugs of person A in yellow and the bugs from person B in blue, and we count up simply the different types of bugs that are there. So you can see in yellow, they have this kind of circular bug, this line of bugs, and then this one with the flagella. In person B, they have more types of microbes represented, and they also have a more even distribution of those microbes. So person B has both greater richness and evenness in their gut than person A. So that's alpha diversity. Now we know that alpha diversity increases dramatically from birth to around 4 years of age, and then it seems to kind of stabilize at an adult-like level from that point forward. Now in adults, alpha diversity is really considered to be better for health, as there's more functional redundancy in the microbes in the gut. But it's not necessarily clear what higher alpha diversity means early in life. So if you've got relatively higher alpha diversity at, say, 2 years of age, what is the functional significance of that? And that really highlights the early stage of this research. We really don't know what the implications are. Now, the next metric that I'm going to talk about today is called differential abundance. And this is also a within individual metric where we look at the relative abundance of bugs that are represented in each person's gut. And we can see that there are 3 bugs that are overrepresented in the sample from the blue individual and not in the yellow individual, and 1 that's overrepresented in the yellow individual and not present in the blue individual. So given their functional roles in producing metabolites, higher abundance of certain microbes in terms of this differential abundance metric may be associated with health outcomes in people. So with those things in mind, let's now ask the question, how does caregiving adversity impact the gut microbiome? And in this particular study, I wanted to highlight some fantastic students of mine, Frank Cadassi and Genesis Flores, who are working on this project together. So in this particular study, what we see is that higher levels of postnatal adversity, and in this case, the postnatal adversity is constituted by higher maternal anxiety and depression symptoms, which are known to affect the children. This was associated with a sharper increase in alpha diversity from birth to 2 years of age. So each individual here has 4 different time points or data points across that first 2 years of life. So postnatal adversity clearly changes the development, the trajectory of change in the gut microbiome. But again, it's not yet clear what implications this has for youth health. Moving beyond 2 years of age, we also see that postnatal adversity exposure changes the trajectories of alpha diversity even into the adolescent period of development. So here I'm going to highlight some data from my graduate student, Nomi Gantz. And here the caregiving related adversity was a little different. Here we're looking at caregiving adversity exposed youth who had been maltreated or were adopted from foster care. So in the comparison group, what we see, so this is our alpha diversity metric, is that from 2 to 17 years of age, there really isn't so much change in alpha diversity. It's fairly stable. This is not a significant slope here within the comparison group. And as I mentioned earlier, that's exactly what we would predict. So there shouldn't be much change in the microbiome across these years under typical conditions. But what we saw within the adversity exposed group was really quite interesting. So here we're seeing that there's a significant non-linear change in the microbiome in terms of alpha diversity within the adversity exposed youth. Specifically, there's lower alpha diversity in the young individuals. It then peaks mid-adolescence and then it decreases in the older adolescent period. So again, these trajectories of development and the way that they're affected by adversities are dynamic across infancy, childhood, and even into adolescence. Now, in this same study, Nomi found non-linear change in the microbiome when looking at the single taxa level. And again, this change was moderated by adversity. So here we're looking at individual taxa. We actually saw 12 microbes that showed an age-related change in relative abundance that was moderated by adversity exposure, so that depended on adversity exposure. But I'm just showing you 3 examples here: Clostridium inocuum, Eubacterium hallei, and one called Butricimonas. Don't worry about these names. It doesn't really matter what these microbes are at this point. It's just important to know that there are at least 12 that showed this age-related change that was moderated by adversity exposure. So here we have age on the x-axis and the microbiome relative abundance on the y-axis, so going up. And the comparison group is in blue, the adversity group is in orange. So the first thing to note about this data set is that the microbiome, even within the comparison group, so the blue individuals, this is changing across age. And that's significant because there is a strong narrative derived from studies of alpha diversity that the microbiome is done developing by around 2-3 years of age. And clearly that's not true of alpha diversity in groups of individuals who've experienced caregiving related adversity in our previous slide. And it's also not true here in terms of the relative abundance of single microbes, either in typical development or after adversity. And that's the second thing to note, which is that adversity changes these developmental trajectories. So now we can see that adversities affecting the caregiving relationship are changing the development of the microbiome at the community level and at the individual taxa level from birth all the way through adolescence. Now comes the next obvious question, which is, why should I care? Or why should you care? Of course, the microbiome is interesting to microbiologists for a variety of reasons, but for people who are interested in mental health, so psychologists, psychiatrists, care providers, researchers, we care about the microbiome because of its links to mental health. So we wanted to see whether any of these microbes out of the 12 that were associated with age and adversity, whether any of those were also associated with mental health outcomes in youth. And what we found was that actually these three, this is why I showed you these 3 of the 12, were all associated with internalizing symptoms in youth, which is anxiety and depression. So what did that look like? Here we have the Clostridium inocuum. So that first microbe that I showed you, it tended to be slightly higher in the adversity exposed youth, at least earlier in development. And it was positively associated with internalizing symptoms. So more of this microbe tended to be worse mental health symptoms. So this suggests that this is potentially, not good for mental health, or is certainly not related to good mental health outcomes. The second one is this Eubacterium hallei. This is an interesting one because it actually produces butyrate, or at least a lot of species within this genus produce butyrate. Butyrate is a potent anti-inflammatory agent. It tends to be positively associated with, good things in terms of brain health and behavior. Yet we're seeing this time, this genus level of this Eubacterium hallei was positively associated with internalizing symptoms. So a little bit of a confusing association there. But finally, the one I wanted to highlight for you and pay most attention to is this one all the way on the right here, Butricimonas, which tended to be a little lower in general across the board in adversity exposed youth. And we found that this Butricimonas seemed to potentially be protective against poor internalizing outcomes. It was negatively associated with internalizing symptoms. So more of this microbe, better mental health. So then the final question comes, so now that we know that these microbes are associated with mental health outcomes, is there anything that we can do about it? Now, many people who are interested in intervening at the level of the microbiome are very interested in things like probiotic interventions. But in fact, diet is a pretty non-invasive and often inexpensive way of altering the microbiome. And it may actually be more tolerable in this developing population of young people. But before we go in and do a diet intervention, we first wanted to look within this data set and see, well, is there any evidence that these types of associations are moderated by the diet that children were consuming? And so interestingly, in this study, NOMI found that fiber in the diet was associated with enrichment of this potentially positive or beneficial microbe in the gut, Butricimonas. And it was so specifically in the adversity exposed group. So specifically, we found in the interpersonal adversity exposed youth in the orange, there was a positive association with the amount of fiber in the diet and this, what we're going to call a potentially protective microbe, Butricimonas. Now, supporting this association, NOMI also found that fiber consumption itself was negatively associated with internalizing symptoms, so anxiety and depression within the adversity exposed group, again, specifically within that group. So this suggests that fiber has unique beneficial relationships with mental health related microbiota in adversity exposed youth. And this really highlights the potential for future nutritional interventions that may have an especially salutary effect for adversity exposed youth. And so we're kind of investigating those pathways now. Now, while the gut microbiome community is clearly the darling of the microbiome world, so if you've ever looked at the New York Times, they've got a story about the gut microbiome every second week, there are actually other microbial communities that exist outside the gut that are linked to mental health and that might be affected by caregiving related adversities. So it's important for us to look and see what those are. So what do we know about the impacts of caregiving related or childhood adversities on the next largest microbiome community in the body, which is the oral microbiome? So there's actually one study which has already shown that the oral microbiome of adults who had experienced childhood adversity was disrupted. And so here in this study, they didn't see any effects at the community level, so no change in that alpha diversity metric or how many bugs are in the gut, but they did see a number of differentially abundant microbes which were associated with prior adversity exposure. So my graduate student, Nomi, was really interested in looking at the impacts of these experiences earlier in development, so looking at the developmental kind of trajectory there. And she also really wanted to zoom in on pathways that intersected with the stress hormone cortisol, as we know that cortisol becomes really dysregulated in the context of early life adversity. And Nomi had this very interesting hypothesis that the oral microbiome of adversity exposed youth may become less sensitive to more recent fluctuations in cortisol. And that's because of studies in immune cells, which had basically shown that. So immune cells become less sensitive to the regulatory effects of cortisol after caregiving adversity or childhood adversity exposure. So in this particular study, we had access to hair cortisol data, which indexes tonic exposure to cortisol over the last few months. So we literally take a sample of hair, we grind it up and can look for cortisol there. And she used that recent cortisol exposure as a moderator in her models where she was examining the association between chronic exposure to adversity from early in life with the oral microbiome composition. And so remarkably, what Nomi found in this study was essentially the same thing that had been shown with immune cells. So within the oral microbiome community, she found that alpha diversity on the y-axis here was negatively associated with recent exposure to hair cortisol within the comparison group, suggesting that within the comparison group, the oral microbiome community was regulated by cortisol or associated with cortisol. And this was not true of the caregiving adversity exposed youth. So here, caregiving adversity means that the oral microbiome doesn't seem to be responsive to more recent fluctuations in cortisol. Now, outside of the influence of cortisol, Nomi in this study also found that several taxa were associated with caregiving adversity, including this pathogenic Porphyromonas species, which was higher in the adversity exposed than the comparison group. And critically, she found that the Porphyromonas species was in turn positively associated with more internalizing symptoms in youth. So again, those anxiety and depression symptoms. So this suggests that even microbial communities outside of the gut, which are affected by caregiving adversity, may play some role in the mental health of youth. And the exciting thing about this is that it really opens up a variety of additional pathways to potentially influence the microbiome and thus indirectly have an impact on youth mental health. And these are even simpler than dietary changes because we can actually change the oral microbiome community through simple oral health behaviors like toothbrushing. So to summarize part 2, what I've shown you is that when you directly experience childhood caregiving adversity, it's associated with changes to the development of children's gut microbiome community composition and the differential abundance of microbes in the gut. And it's also associated with the way that the oral microbiome responds to cortisol or recent fluctuations in cortisol, as well as the differential abundance of microbes in the mouth. And some of these microbiome outcomes, both in the gut as well as in the oral cavity, are associated with youth mental health. But one issue, of course, remains, and that is how. So how are these microbes in our guts and in our mouths influencing something as complex as mental health outcomes? And of course, there are a multitude of ways that this could happen, but really all of them involve the brain. So now we have a bit of a problem. We are trying to link two incredibly rich, complex multivariate organs, the brain and the gut microbiome, with one another. And we want to do it in a way that tells us something meaningful about a heterogeneous, non-distinct clinical symptom cluster like internalizing symptoms. And we want to do all of this, moreover, non-invasively in small humans who don't like to lay still in an MRI scanner. So how are we ever going to make progress on this front? It's a pickle. So I want to show you today our first attempt using a top-down multivariate and longitudinal approach. So what does that mean? Well, here we had data on 55 individuals who had all of these data. We had a microbiome sample from them when they were 2 years of age. We had high quality resting state data when they were in the MRI scanner. And we were looking at inter and intra connectivity. So meaning connectivity within networks that are defined and then between different networks in the brain. And then finally, we had internalizing symptoms in these youth, so again, anxiety and depression, when they were 7.5 years of age. Now, before I go into the data, I wanted to acknowledge where these wonderful data originate from. So we've been really fortunate to collaborate with the group behind this incredible birth cohort study called Growing Up in Singapore Towards Healthy Outcomes, or GUSTO. It's the largest of its kind in Singapore, and it has collected densely sampled data from the prenatal period all the way through 10 years of age in the most current data collection effort. And this team has been really generous in allowing us to work with these data. And I just think that it deserves special attention because it greatly accelerates the pace of research and discovery. And it really diversifies microbiome science and neuroscience, which tends to concentrate mostly on samples from the United States. So I'm going to show you some data from this cohort now, and then more of it will come up later in the talk. So in this particular study, this was headed by my graduate student, Frank Hadassi, and a postdoc or former postdoc, Dr. Jessica Wee. We had a 3-step approach. So in the first step, we used an approach, it's basically a machine learning or a data-driven approach, to ask what combination of brain networks best explains variability in internalizing symptoms. And we're going to call that a brain signature. Whatever pops out is our brain signature. And we're then going to use that brain signature as the dependent variable or the outcome in step 2. And now we're going to do the same thing, but now with gut microbes. So what combination of gut microbes best predicts variability in these internalizing associated brain signatures? And then in our final step, we want to then link it back to internalizing symptoms. So we're going to take the microbiome component from step 2, the brain component from step 1, and now we're going to ask the question, is the microbiome indirectly associated with youth internalizing symptoms via these internalizing associated brain networks? And so we did this, we found lots of interesting things. I'm just going to show you the most interesting data here for the sake of time. So in step 1, we found that this linear combination of resting state brain network connectivity patterns explained most variance, or maximal variance, I should say, in internalizing symptoms. And this was really characterized by connectivity between the SOFA network and a variety of other brain networks. So the SOFA is the striatal, orbitofrontal, amygdala network. And for those of you who've dabbled in neuroscience or maybe neuroscientists yourself, this will not come as a surprise. So these subcortical and cortical brain regions tend to show up in a lot of affect or emotion-related studies. And so this SOFA network was connected with a range of other networks in the brain, which was not surprising. So things like the default mode network and the ventral attention network. So again, pretty sensical brain signature to emerge here. So then in the next step, we wanted to ask that question, well, what combination of microbes predicts variability in that SOFA signature? And here is where things got really interesting. So the first thing to note is that we got high positive loadings from Veillonella and Intestinibacter. So more of these microbes was associated with variability in this network. And these 2 microbes have in prior literature been associated with intestinal inflammation and depression in adults. We also saw high negative loadings from Fusocatinibacter and Catinibacterium. And these have been negatively associated with anxiety, depression, and physical illnesses in adults in the prior literature. So this was really exciting to see some microbes emerging from this pattern that were linked with mental health in prior literature. And so then finally in step 3, we tested that, what's called a mediation, so the indirect effect. And we did see support for an indirect effect. So basically, the SOFA microbiome pattern was associated with increased internalizing problems when youth were 7.5 years of age, via effects on the, or associations with the SOFA signature when youth were 6 years of age. And moreover, this mediation was really specific to this pattern, so both these brain networks and this microbiome pattern. So I didn't show you anything else for the sake of time, but it's actually specific to this pathway. So now to finally summarize this section of the talk, I've shown you that directly experienced childhood caregiving adversity impacts both gut and oral microbiome communities, some of which are associated with mental health outcomes in youth. And now in the last part of this section, I've shown you that we can work backwards from youth mental health symptoms to identify relevant brain networks and then microbes that are associated with those brain networks. And this is important because we can then identify microbes that might be potential targets from therapy. So we really think that taking this backwards approach, so working backwards from the internalizing symptoms that we care about through the brain networks and then ultimately to the microbes, might be a really good pathway to use in order to get to these outcomes. So in the next part of the talk, in part 3, I'm going to now take this intergenerational focus. So I want to be looking at how early caregiving adversity in generation 1 can influence outcomes in the next generation. So these are the children of exposed individuals. And so we'll be focusing first on influences at the level of the brain, and then we'll integrate this focus on the wider brain-gut microbiome axis. So again, focusing first on brain and behavior. In this study led by my former postdoc, Dr. Jessica Wee, we had data on 541 mother-child dyads from that wider GUSTO study. And these dyads had data from the following data points. So we had mums reporting on their own experiences with childhood trauma, and they reported on these while they were pregnant, but these are childhood trauma experiences. We then had mums report on their own mental health problems during the prenatal period and the postnatal period, which can be considered to be adverse events for the developing fetus and the young child. And then finally, we had children's report of emotional health problems themselves. So they self-reported, and then we also had parents proxy report for their child when the child was 7 years of age. So what we found in this study was that maternal childhood trauma was related to worse parent proxy reported child emotional health outcomes at 7 years of age, and that this occurred serially through a pathway of worsened maternal prenatal and postnatal mental health in the context of high levels of maternal childhood trauma. We also saw that maternal childhood trauma was related to worse parent proxy child self-reported emotional health outcomes, and that this occurred through a pathway of worsened maternal prenatal mental health. Now, in a subset of that wider data set where they had neuroimaging data on youth, we had high quality resting state data from 89 children who were 6 years of age at the time of the brain scan. And so just look to see whether any of these relationships between any of these experiences of adversity and amygdala prefrontal cortex resting state functional connectivity when youth was 6 years of age. And we wanted to focus on amygdala prefrontal cortex connectivity because it's been examined extensively in relation to emotional health and the development of self-regulation in youth. And so what Jess found was that over and above maternal childhood maltreatment or maternal prenatal health, maternal postnatal mental health was uniquely associated with lower or more negative amygdala prefrontal cortex functional connectivity in second generation youth when they were 6 years of age. In turn, she found that lower amygdala prefrontal cortex resting state functional connectivity when youth was 6 was related to worse parent proxy reported and child self-reported emotional health problems when youth was 7 years of age. So this suggests that there really are intergenerational impacts of adversity on children's emotional health and brain function, and that at least for the behavioural effects, so when we're looking at child emotional health outcomes, these seem to be mediated through maternal mental health in the prenatal and in the postnatal period. But when we're looking at children's brain health, these seem to be more closely tied to the most proximal relationship to the child, which is the mum's mental health since the child's birth. Now, again, picking up on this idea of these transdiagnostic mechanisms, so revisiting this wider brain-gut axis, my graduate student, Fran, was really interested in using this same cohort to understand whether there might be an impact of maternal childhood adversity, prenatal and postnatal mental health on other features of the child's biology, like their gut microbiome, all the way down at the other end of the brain-gut axis. So we had these data in 450 youth from that wider sample. And here, Fran is controlling for things that impact the microbiome, like diet, ethnicity, sex, duration of breastfeeding, etc. So here what I'm showing you are some of the data from that study. We have differentially abundant microbes plotted, with those on the right of this line here higher with more adversity exposure, and those on the left of the line higher with less adversity exposure. So the first thing to note is preconception adversity. So this is maternal childhood maltreatment. This was associated with a higher abundance of this microbe down the bottom here, Clostridium sensu stricto, which is actually a butyrate producer, but it seems to be maybe an inefficient butyrate producer that should have probably been outcompeted by around 2 years of age by more efficient butyrate producers in the gut. So this hints that butyrate production may potentially be less effective in second generation youth who have mums reporting high levels of childhood trauma exposure. Next, in terms of prenatal adversity, we found 2 types of streptococcus in pink were higher in the context of prenatal adversity. This is part of the normal microbiome, but several species are pathogenic, which may explain some of that association. And then we saw lower levels of ruminococcus. And it's hard to interpret this because ruminococcus has been associated with immunity, both anti and pro-inflammatory, as well as dopamine within the context of Parkinson's disease. And then finally, postnatal adversity, so high maternal mental health symptoms from birth to 2 years of age, as well as stressful things that have happened to the child from birth to 2 years of age. This was associated with a lower abundance of Parabacteroides, which is a potential probiotic, and a higher abundance of Finagoldia, which is an opportunistic pathogen. So based on these data, and in the context of some of our earlier findings, we're again seeing that butyrate production in the gut is something that seems to be affected by adversity exposure. And we're also seeing immune function crop up as something that may be associated with adversity exposure, especially in a generational context. Critically, you're going to see a lot of hand waving and may, using some qualifiers here, and that's on purpose. And that's because it's really hard to tell what the functions of these gut microbes are. And that's simply because the resolution of these data is not so great. So here we're looking at the genus. So let's revisit some taxonomy from maybe high school is the last time you might have thought of this. So here's a taxonomy chart of domestic dogs. So if you take the domestic dog, we can see that it is in the genus Canis, which it shares with species as diverse as jackals, wolves, and coyotes, before it then gets to its own species, which is, Canis lupus familiaris. So when we sequence bacterial DNA, depending on the approach, we're going to get different levels of resolution, all the way down from the kingdom, all the way down to the species, strain, and even substrain level. But as you can see, the type of sequencing approach that we have used in all of these studies, which is called 16S rRNA sequencing, it really only gets us down to the genus level. And as I just demonstrated, there's tremendous variability within a genus. This is in dogs, but the same is true of bacteria. So in order to get down to the species or subspecies or strain level, we really need more high resolution approaches like shotgun metagenomics and other sequencing approaches. And this is going to help us learn a lot more about the microbiome. Now, the primary reason that we don't have a lot of data using these approaches is simply because they're orders of magnitude more expensive. And I would say that within the psychology and psychiatry field, gut microbiome research is really still in its early childhood phase of development. So there's really a lot that we're needing to learn. We need to get a lot more funding within the field specifically to get at these higher levels of resolution. Now, thankfully, in my lab, we have some of that data in hand now, which is getting us closer to understanding the functions of the gut that may be affected by adversity, but unfortunately, it's too early to share those data right now. So just to note that future microbiome research is really going to need funding that supports shotgun sequencing performed in this kind of multi-omics framework. So not only understanding what bugs are in the gut, what do they have the potential of doing, what do they actually do, what do they produce, what genes are transcribed. There's just so much more information that we can get from these data and that we need to get from these data in the future. So, summary of part 3, what I've shown you is that maternal childhood adversity seems to be associated with children's brain and behavior, the impacts on maternal prenatal and postnatal mental health. So second generation outcomes, first generation impacts, and then it seems to be mediated through this mother. We've also shown you that children's microbiome seems to be affected via maternal childhood adversity exposure, and here the pathways somewhat overlap with those of prenatal and postnatal maternal mental health problems. So given these findings and the clear importance of maternal mental health in the intergenerational impacts of adversity, we're becoming increasingly interested in the mother, particularly during pregnancy, as a conduit for some of these intergenerational adversity effects. And so in the last small part of this talk, I'm going to show you some very recent data we've been collecting, looking at caregiving adversity impacts on the transition to parenthood. And here I'm going to be focusing not on the gut microbiome, but on another mechanism that involves integration of the brain and the body, interoception. All right, so pregnancy is a critical period of development, which is characterized by heightened risk for depressive symptoms. Up to 12% of women will experience depression during their second and third trimester, and those rates have even increased post-COVID. And critically, depression during pregnancy is further amplified by pre-existing environmental vulnerabilities like childhood trauma or adversity exposure. Now, this is important not just for the health of the mother, which is of course reason enough, but it's very important because it can also perpetuate, peripartum depression can perpetuate cycles of adversity across generations. Now, it's been convincingly argued by many research groups that depression at other stages of the lifespan arises from a process of disordered interoception. Interoception is the way that the brain perceives, integrates, and models sensory information which is generated from the body. Now, while some differences exist between depression at other stages of the lifespan and depression which occurs in the perinatal period, both are characterized by that core negative affective symptomology involving like low or irritable mood, as well as abnormalities and altered functioning across numerous bodily systems, including the brain, immune, and neuroendocrine systems. And it's this widespread impact of depression on the entire body that points to the idea that there may be inefficient energy regulation occurring in depression, which may in part be responsible for the negative affective symptoms. Now, critically, energy regulation is supported by that process of interoception, and so it won't come as a surprise to you to note that interoception has been impaired in depression in numerous different studies outside of pregnancy. So here we wanted to see whether early life adversity exposure impacted the way that interoception changed during the pregnancy period. So here I'm showing you some data from a theoretical model developed by myself and my student Paul Savoca, where we're basically looking at or hypothesizing about how interoception should change across pregnancy. And one thing we think for certain will happen is that interoception should change across pregnancy, because there's really no other stage of the life, bar from early infancy, where we see such massive changes in the sensory signals that are generated from the body. So this is a really compressed period of development in adulthood. So this conceptual figure shows how metabolic demands are drastically increased during pregnancy. And what we theorize is that under normal circumstances, so no early life stress, or under typical or optimal circumstances, let's say, interoception should really become boosted in pregnancy to keep up with these metabolic demands, and that this may actually provide some resilience against depression. Now, interestingly, studies have shown that adversity exposure is associated outside of pregnancy with lower levels of interoception. And so our hypothesis is that this may actually become amplified during pregnancy, leading to an increased risk for major depressive disorder, specifically within that group. Now, Paul has since found support for these hypotheses. So here we're looking at interoceptive sensibility, which is a self-reported measure of interoception. And he found early life adversity moderated differences between pregnant and comparison groups on 3 subscales of this Maya interoception sensibility questionnaire. And this was moderated by adversity exposure. So here you can see in the yellow, these are called regions of significance. So this is really where we're seeing the differences between the groups. So what he sees is that for people with lower early life adversity exposure, so here on the left panels of B and C, pregnancy boosted interoceptive sensibility on the self-regulation and the noticing subscales. So pregnant individuals with lower early life adversity, more so than non-pregnant individuals, reported higher ability to regulate themselves through attention to their body signals, and they also notice more of those body signals. But for individuals with high adversity exposure, pregnancy was associated with lower scores on the attention regulation and the noticing subscales. So suggesting that we see both buffering as well as impairing effects of pregnancy on interoception that are toggled by adversity exposure. Now, critically, we see across both of these measures, self-regulation and attention regulation, that they are negatively associated with depression symptoms in both groups. So this suggests that interoception may impart a buffering effect against depression, which is boosted in pregnancy for individuals with low adversity exposure and weakened for individuals with high adversity exposure. So to summarize part 2, maternal childhood adversity seems to impact mothers' experiences of interoception changes during pregnancy in ways that are important for maternal mental health. And this is actually a really important set of findings because focusing on interoception during pregnancy may actually be an important way to address intergenerational patterns of trauma, because interoception can be targeted through treatments like certain mindfulness-based therapies, as well as interoception training. So returning to our research priorities that I noted at the start of this talk, transdiagnostic mechanisms, appreciating the importance of generational experiences. What we think might be really important within these research themes is to try and focus on features of an individual's biology and psychology that integrate brain and body. So that is focusing on the brain-gut microbiome axis and then focusing on interoception. And we also think that focusing on a mum's mental health and adjustment to the metabolic strain of parenting, both of these together are going to have or go a long way in helping us to improve individual well-being after adversity exposure. Now, of course, there's much more to do on this front, and of course, I look forward to sharing those data with you sometime in the future. So I wanted to end by acknowledging the fantastic students in my lab that make all the difference when it comes to research. So Frank Cadassi, Nomi Gantz, Jessica Wee, Paul Savoca, and Genesis Flores. There are more students in the lab. These are just the ones that contributed mostly to the data that I presented today. And a wider thank you to the students, postdoc, and staff, the collaborators, and our funders, including, very importantly for this talk, the Brain Behavior Research Foundation. And thank you all for paying attention.
Well, Bridget, I want to thank you for a wonderful presentation, packed full of knowledge and information, really just extraordinary. So thank you for that. We don't really have time now for questions, but you covered so much. And I just want to point out that I think this is an example of research that has the potential to have tremendous impact, not only for treatment, but for prevention. And so much of our work in the past in the field of psychiatry and brain research hasn't yet focused on prevention, but now we are, and this is an example of that. So thank you very, very much for the work you're doing, the work you're continuing to do, and for this presentation. And we will want to have you back again to share more information as it develops.
That's so great. Thank you so much. I really appreciate it. And thank you all for coming along and hearing what we're doing.
Good. I also want to thank our audience today for joining us. And I'd like to ask people to please consider making a donation to BBRF today. 100% of every dollar donated for research is invested in our research grants. We're able to do this because our operating expenses are covered by separate foundation grants. This means that when you donate $1 for research, that dollar goes directly to the scientists To make a gift, please visit our website, bbrfoundation.org, or call us at 1-800-829-8289. I'd like to tell you about another event that is coming up on December 4th, our educational webinar entitled ADHD, What You Need to Know, a Q&A with Dr. Stephen Hinshaw. You can see this information right up here right now on the screen, and more information. You can get more information about it at our website. I also want to invite people to please join us again on December 10th at 2 p.m. Eastern Time, when Dr. Catherine Brownstein, an associate professor in pediatrics at Harvard Medical School and a research associate in the Division of Genetics and Genomics at Boston Children's Hospital, will present Investigating the Genetic and Biological Mechanisms that Predispose to Early Onset Psychotic Illness. I want to thank everybody again, and remember, together we can dramatically improve the lives of those living with mental illness and enable more people to live full, happy, and productive lives. Take care.